I will separate the topics perhaps even further in this case. So I mostly in the last 10 minutes or so, I was mostly focusing on perineuronal nets in general as an extracellular matrix aggregate structure. But as I said very early on, so extracellular matrix is not just something like that. It will also have something that is floating fluidly in general in the extracell between cells. And I think we need to classify them quite clearly. So I normally call the other ones the more mobile one as the loose extracellular matrix, and the other ones are aggregated matrix like perineuronal nets. During development, we do not have perineuronal nets. Development here, I'm referring to the embryonic stage in the nervous system development. There's no perineuronal nets there. And what you have is basically these extracellular matrix molecules, loose extracellular matrix between cells. And the reason why it is you're correct, you're actually suggesting that. So during development, the extracellular matrix is very important because at that time, the network of the nervous system has not been established. Everything was basically based on diffusion. So it's diffusion of molecules creating gradients, allowing molecules, cells to grow in a particular direction, having a directional growth. And because of these extracellular matrices actually are organized as a hydrogel system, and they will have ability in retaining molecules in a certain levels and a certain gradients as well. And from this gradient, allowing molecules and growth cones and cell movements to happen. And that is the reason why if you disturb the system during development, you actually have a completely misdeveloped nervous system, and of course, then it will affect a lot of the growth afterwards. Mm-hmm. And that is the reason why the extracellular matrix during development is super important. So they allow the gradient to form morphogens. I think when people are thinking about neurodevelopment, they think about morphogens a lot. So, for example, hedgehog and different molecules related to that. But these molecules, if you put something in a bowl of water, they will just diffuse everywhere. Yeah. So, and then at one point they will be mixed completely, even concentration everywhere. This is not the case in the development. You will have this gradient formed, and they would be maintained. And it is this maintenance of the molecular concentration allowing developments to happen. And extracellular matrix is the one holding this principle of retention of this concentration. You may ask the question how does extracellular matrix actually affecting this? It's because of the weak interactions that these molecules interact with the matrix molecules, allowing them to have a preferential or differential interactions. And that is how the gradients are formed and maintained in general.